All right, all, we're going to continue on with our AP Biology exam review. Right now, we are going to do Unit 5, which is focused on inheritance. So this is where all the genetics lives. So the first thing that we're going to talk about is meiosis and how kind of sexual life cycles occur. In the previous Unit 4 review, we talked about mitosis, so how cells just go on living their happy lives, continuing to divide with the purpose of growing, but our sex cells do meiosis, so that's where we are here. In terms of reproduction, there are two options. On the left, you have asexual, so basically it's just cloning. You are producing one genetically identical copy from a single parent. So things that do this, bacteria are one common example. A lot of prokaryotes do this. The problem is it doesn't really create any variation in the population. There's no enhanced genetic diversity because you're literally just cloning. It happens really fast because you only need one parent, and you can divide fairly quickly by either like budding or binary fission. But again, you're lacking diversity. On the right, we have sexual reproduction. So this does require two parents. And don't get me wrong, plants, for example, they can kind of fertilize. The male part can fertilize the female part of the same plant, yes. But technically, it has to have two parents. And it produces sex cells called gametes. And those are what come together to make an organism. There's a lot of variation involved because when you have different individuals, for example, they're going to bring different genetics to the table. And when they combine, then you get unique combinations. It's slower, and it does require a lot more energy, but sometimes the enhanced diversity makes it worthwhile. So thinking about things that do this, plants and animals. And now begins the onslaught of vocabulary. So in terms of cells, the ones that typically just go through mitosis are not sex cells, are called somatic cells. These are your body cells. Somato means body. We already learned humans have 23 pairs, so a total of 46 chromosomes. And we are diploid, so that is our 2N number, two sets, 23 from mom, 23 from dad. And those are called homologous chromosomes. So you have one chromosome 1 from mom, one chromosome 1 from dad. They're both chromosome 1s. They both have all the same genes on them. They might be different versions, but they are all of the same. So they are called homologous chromosomes. And that's true for all of the different pairs that you have. Autosomes are the 22 pairs that are not sex chromosomes. So in total, we have 23 pairs. 22, numbers 1 to 22, are all just our normal chromosomes that determine everything except for sex. The last pair of chromosomes, our 23rd pair, are sex chromosomes. And in this case, there are two, one from mom and one from dad. You can either have two X's and be a female biologically, or you can have an X and a Y and be a male biologically. Our sex cells, or our gametes, have N, meaning haploid. They have half the genetic material, which makes sense because when an egg and a sperm comes together, you combine those two sets, and that's how we get up to a total of 46 chromosomes. So in each one of those gametes, there's 1 to 22 individual autosomes and then the one sex chromosome. So eggs always have X chromosomes. Moms only give children their X chromosome. In terms of the sperm, males determine sex because those sperm will either contain an X chromosome or they could contain a Y chromosome. So here's just showing you what homologous chromosomes might look like. In this case, there is a total of 6 chromosomes. So they're color-coded. You have 3 homologous pairs, a total of 6 chromosomes. And it's a little tricky because in this particular picture, this would have been in after S phase, where the chromosomes have copied themselves, but before they've gone through mitosis because they haven't split yet. So this is technically one chromosome, and it has its sister chromatids, and its homologous chromosome would be the one right across the way that's of equal size. And that's kind of what it's showing you here too, down with the middle-sized ones, that those are homologous chromosomes with their duplicated chromatids next to each other. It's a lot of tricky vocabulary. We can use karyotypes, which is basically a photo of all of your chromosomes lined up next to each other, to easily see genetic disorders. For example, in this picture, we have Down syndrome, which means they have an extra 21st chromosome. You can see down here there's 3. In terms of our chromosomes, the first chromosome is the longest. That's kind of how it worked out. They made the longest one number 1, and then it's kind of decreasing in size all the way to 22. And then we have our sex chromosomes here. So this would be an example of a boy. If it had been a girl, there would be 2 X's, and then there would be nothing over here by the Y. Now, in terms of life cycles, it kind of oscillates back and forth. So fertilization is when the egg and the sperm come together, or the gametes come together and create a zygote that is diploid or has the full 2 sets of chromosomes. And then those organisms create new sex cells by meiosis, which basically means they take that 2N or double set and cut it in half and create sex cells that have just 1 set. So that's why it's called kind of alternate life cycles. We cut the genetic material in half when we make our sex cells, and then they come together to fertilize and make a new organism. Then we make more sex cells, more organisms, and so on and so forth. Once the zygote is made, then everything that's not a sex cell basically goes through mitosis to just grow and develop. Different organisms do it a little bit differently. So we are on the left in the animal category. Like I mentioned, we make our gametes, which are our sex cells, and then we get one from someone else because we do sexual reproduction, gets fertilized. Voila, we have a diploid organism that grows up and then makes its own sex cells, and so on and so forth. Plants do it a little bit differently. They actually have both N organisms and 2N organisms. So haploid organisms are called gametophytes, and they just got to go through mitosis. They can become fertilized and become sporophytes, but they kind of have an alternation of generations there as well, with alternating between haploid and diploid organisms. Fungi and protists kind of do a little bit simpler thing in terms of us. They have gametes that come together via fertilization. They have a zygote that's 2N, and then they go through meiosis and so on and so forth. So there is that alteration between N and 2N. Meiosis is basically cell division twice, because if you do it 2 times, then you are able to successfully cut the number of chromosomes that you have in half. So also, because you're dividing twice, you generate 4 daughter cells, and those 4 daughter cells will then have half of the genetic material that you started with. So for example here, this one has 2 chromosomes, 1 homologous pair. Then when it goes through S phase and copies its DNA, then you have 2 sister chromatids. Meiosis 1's job is to separate homologous chromosomes. So you'd end up with 1 chromosome 1 in one cell and 1 chromosome 1 in the other. Then meiosis 2 is just like mitosis. They all line up in the center and they split, and you end up with 1 chromatid in one cell and 1 in the other. When they split, they become their own chromosomes. So you end up with 4 daughter cells. Each one has half the genetic material as the parent. So the parents had 2 in this case. The daughter cells only have 1. So that's the super quick nutshell version of meiosis. All right, so just kind of running through what's kind of occurring. I don't think you need to know the super specific details, but as I mentioned, it's a lot like mitosis, except there are a couple small extra things that are occurring in meiosis that really enhance genetic diversity. So in interphase, chromosomes are still copied in that S part of interphase that we've learned about before. In prophase 1, this is in particular where homologous chromosomes line themselves up. So in mitosis, they just all line up in one line, but for meiosis 1, they line up as homologous pairs. So this would be like chromosome 1, 2, 3, so on. That grouping where both homologous chromosomes are right next to each other, so you have a total of 4 sister chromatids, that's called a tetrad. Tetra means 4. And this is unique because this is where crossing over takes place. So sometimes what happens is a chunk of one of the chromatids on one homologous pair can actually swap places with a chunk of a chromatid on the other homologous pair. And that crossing over really allows new and unique combinations of genetic material because the homologous pairs are swapping. So like a chunk of moms goes to a chunk of dads. And so that's a different combination than you would have ever, had before. Then in metaphase, they all line up down the middle. In anaphase, they split. And in telophase and cytokinesis, they become 2 cells. So effectively after meiosis 1, you have cut the genetic material in half because you've separated homologous chromosomes. So the resulting set has half of what they had before. The key difference here is, again, in prophase 1, that idea of crossing over. So that creates what's called a chiasmata or chiasmata. So that's where the crossing over actually takes place between the tetrads. They line up, they separate, voila. We now have haploid cells. Meiosis 2 is essentially mitosis. You have one set of chromosomes per cell. So what happens is, the nucleus disappears. There's no interphase. So I guess that's a hallmark. It doesn't really go through the full cell cycle. There's no crossing over like there was before. They just kind of line up, separate, and divide. And so you can kind of see here, they're lining up, they're separating, and then they divide. So your resulting product ends up being 4 total cells because that happens with the one product of meiosis 1 and then the other. So you end up with 4 cells that have half the genetic material as the parents, and they have unique combinations thanks to crossing over. Just to kind of recap, meiosis has crossing over, mitosis does not. Meiosis has homologous pairs lining up, mitosis does not. And then anaphase 1 is where the homologous pairs separate, but the sister chromatids are still attached. That's not something that happens in mitosis. In terms of genetic variation, I mentioned we can increase genetic diversity with crossing over to get new and unique combinations. Other things to think about, we get increased genetic variation when we independently assort chromosomes. What that means is when the homologous pairs are lining up, they do so randomly. So in this instance, you could have like all dads on the left, all moms on the right, but it doesn't always happen that way. All 23 pairs kind of line up at random along the center. So when they separate, you end up with different combinations that will then result in the daughter cells at the end. Another source of variation is the idea of sexual reproduction in general. When you have a sperm and an egg coming together from 2 different sources, those sources have different sets of genetic material. So that can then bring new and different diversity. Okay, so here's the quick difference between the two. Mitosis is for body cells versus meiosis sex cells. 1 division versus 2. Mitosis makes 2 cells, meiosis makes 4. Mitosis is making exact copies, or mitosis is. Meiosis is making unique copies. Mitosis is basically from when you're a zygote to the time you die. Meiosis is specifically for sex cells. whereas mitosis is for growth and repair. Mitosis does not have crossing over, and meiosis does. Here's the graphic that kind of goes with that. And another table that goes with it. So if you want to read through, go ahead and pause and then kind of read through this. The first vocab to kind of learn about are alleles. And we talked about chromosomes and homologous chromosomes. And as I said, you get one chromosome 1 from mom, one chromosome 1 from dad. And at specific locations on those chromosomes, excuse me, are genes. And it's known that along those chromosomes, genes are in a defined location, but different people have potentially different versions of that gene, and enter dominance, recessive, and so on. So at any particular location, your allele is your version of whatever that is. And you as a person, because you have one chromosome from mom and one chromosome from dad, you get one allele from each, and that will generate your genotype that we'll see in a minute. So Mendel, our monk with the peas, came up with a couple different principles or laws. He kind of came up with the idea that there were alternate versions of genes or alleles, and that gives us variation in our characteristics. And you get one from each parent. And if for some reason those are different, that one of them will be dominant to the other. The dominant is the one that you actually see outwardly, whereas the recessive one is there, but you can't actually see it in terms of appearance. It's masked by the dominant one. The law of segregation is one thing that he developed. So it's that kind of random assortment piece of two alleles separate during gamete formation. So during the process of meiosis, the chromosomes themselves, and therefore the alleles, segregate or separate from each other when those eggs and sperm are created. So here's just kind of a picture representation of that. If you think about purple flowers have a capital and a lowercase. So they have two different alleles in this particular case, and we'll learn that those are heterozygous. Well, half of their eggs or sperm might get the big P, and half of their egg and sperm might get the little p. So that's kind of the idea of segregation, and they do this randomly. As I mentioned, dominant is the one that you would outwardly see. So we always give it a capital letter. Recessive always gets a lowercase letter. If you have two of the same, homo means same, so homozygous would mean that you either have two capitals or two lowercases. In this case, it would be homozygous dominant if you had two capitals, or homozygous recessive if you had two lowercase. Hetero means different, so heterozygous means you have one of each. And you can see down at the bottom that we then break this up into genotype and phenotype. Genotype are your genetics. So it's the two-letter combination that you happen to have. Whereas phenotype, think physical, it's the physical appearance of those genes. So in this case, purple color is dominant in this flower and white color is recessive. So if there's a capital P, whether it be 1 or 2, you're going to end up with a purple color versus a white color. It's important to know too that your ratios, kind of seen at the bottom there, the ratios can be different across genotype and phenotype, because in this case, the genotypes of capital P and the genotype of capital P lowercase p both yield the same phenotype. So they do end up sometimes being different. And this just rehashes those two vocabulary terms for you. Okay, enter Punnett squares, and there really isn't one on the center of the screen, so that doesn't really help, but we will encounter them as we continue. It's basically a way of coming up with a probability that you will have specific offspring, and I think this right here at the bottom is one example of what that looks like. So you would put one parent's genotype across the top, the other parent's genotype across the side. It does not really matter which one goes where. And then you just kind of line them up in terms of the combinations that you would get. And then you can determine what the genotype and phenotype probability or frequency would be if you were ever to cross those two parents. So this is one clear example of what a Punnett square would look like. Okay. Another concept to think about is something called a test cross. So there are instances where we might have a plant, or we can see the dominant feature in something. Some organism is showing the dominant phenotype, but we can't always tell if it's a homozygous dominant or if it's heterozygous, because physically they look exactly the same. So if we want to determine the genetics behind it, we can do something called a test cross. And the idea is you just take your unknown plant with an unknown genotype and you cross it with the recessive, because we know what the recessive is. In order to even see the recessive phenotype, it must be lowercase, lowercase. And then you just kind of look to see what the offspring produce. If the offspring only produce the dominant phenotype, well, then you know the parent had to be homozygous dominant. If you start to see some recessive individuals in the offspring, then you know that the parent must have been heterozygous, because that's the only way that you can get that combination. So that's called a test cross. It might come up in experiments presented to you. Another concept that Mendel came up with is the law of independent assortment. So basically, the alleles, or the different versions of traits that you can potentially get, assort independently of one another. They're not dependent, meaning that in this case, P, shape, and color are not relying on each other. So those two traits assort independently. They shuffle themselves up into gametes, the egg and the sperm, on their own, independently of one another. There's two kinds of Punnett squares that you can have. The one that I had showed you previously is called a mono-hybrid cross. It's just 4 squares. That is where you just look at one trait, one character between 2 organisms to see what the probability is. You can also start to look at 2 characters or 2 traits like shape and color, and that's what we call a dihybrid cross. And that generates a 16 square Punnett square. Just kind of backtracking into probability, the idea is that if you want 2 events to occur at the same time, so for example, if I'm looking at P, shape, and color, and I want it to be round and I want it to be yellow, I need those 2 traits to happen at the same time. I kind of multiply those chances together. Similar if I'm rolling some die and I want to get 2 6s. Well, the chance of me getting 1 6 on a dice is 1/6. And then the chance of the second dice being a 6 is 1 in 6 chance. If I need them to happen both together at the same time, it's harder. So I need to multiply their probabilities together. If I'm looking at traits, instead of drawing out an insanely large Punnett square, you can multiply the probability of each one separately. So if I look at just the A's, what are the chances of those A's and those A's making the offspring A's? 1/2. Looking at the parents' B's, what's the percentage of getting that particular B combo in the offspring? 1/2. And then the C's, 1/4. So if I need all of those things to happen at the same time, I multiply their probabilities together. Now, if it doesn't really matter, if it's kind of an or situation, not an and, it doesn't really matter the order, or you're good with having one or the other option. If I want a yellow one or a wrinkled one, doesn't matter, those kinds of things. Then you add their probability, because it actually becomes easier because you're allowing yourself more options. Now, Mendel only kind of thought about strict dominance and recessive. That's the pure normal genetics, we'll say. But there are exceptions to the rule. So there's something called codominance, which is where it's not just straight up dominant and recessive. And there's incomplete dominance and things like that we'll get to. So Mendel was the idea of complete dominance. One completely masks the other. Incomplete means that the one allele is not fully masking of the recessive. So in this case, you might have a red flower and a white flower, and you're going to get a pink flower because for whatever reason, the red allele is not fully covering of the white allele. So this is incomplete dominance. It's not quite fully dominant. So you end up with something kind of in between. And that's easy to then see, knowing those would be heterozygous. And the notation for it's a little bit different because you actually have two different letters instead of just one letter. Codominance is when there are alleles that are equally dominant to each other. So they're not going to mask one or the other. They're both equally expressed. So roan cows are one example where if you have red and white hairs, you actually have red and white hairs. You don't end up with this medium in between pinkish color. There, if you zoom in, there's actually red hairs and white hairs. Another example of codominance are blood groups, because A and B are actually codominant with one another. And it's interesting because it's also an example of multiple alleles. There are instances in life where certain traits have more than two alleles. In this case, there's an A, there's a B, and those two are codominant with one another. Oftentimes they get the notation of capital I with a A or B superscript because they are codominant. And then there's little I, which is the recessive. So there's three alleles for this particular trait. And depending on your combination, you're going to end up with a different blood type. So if you end up having an A and a B, they are codominant. You will see both of them. And both A and B are dominant to the little I, which if you only have those, we say you have type O blood. Here's just kind of a graphic showing you that if you want to pause for a second and kind of analyze. There's also a really interesting idea of polygenetic inheritance. So not everything is determined by one singular gene and your version. Think about all of the traits that people have that are kind of on a continuum or a gradient of some kind. So height is one perfect example. It's not tall or short. There are, huge variations. Same thing with skin color. And so the idea is that there are multiple genes that when you add that combination together, all the different versions have a little bit of impact, and that unique combination ends up yielding the specific skin tone that you might have. So here it's just kind of showing you if skin color being controlled by 8 different genes and your versions of those different genes, then you can end up with, 64 different skin tones. On top of the actual genetics, it's really interesting to note that environment can play a role too, because depending on certain nutrients in the surrounding areas, you can actually then yield different phenotypes. So, for example, these plants have identical genetics and they are just grown in different environments. And because of that, they then yield different phenotypes. That's kind of the nature versus nurture debate. Again, like if you took identical twins like these plants and you put them in different environments, they might end up looking differently or acting differently, even though the underlying genetics is exactly the same. So environment can have an influence. Genetics is not the end-all be-all to the physical appearance of something. When we're looking at how things are inherited in humans, we can oftentimes create a pedigree. A pedigree is basically just a family tree that shows you who might have inherited a particular trait over time. And you can see the parents and the offspring in multiple generations. All right, so this is just kind of a review of all the different types of inheritance that you can see. So take a moment and pause, look at the examples. Same thing here. Go ahead, take a pause, look at the examples. So the last concept in here is the chromosomal basis of inheritance. And the idea here is kind of repeating what we learned before. Genes do have specific locations. In this case, you can see that one specific gene is highlighted in yellow in the picture, and you can see it's on 2 chromosomes because those would be the homologous pair. These chromosomes separate and assort independently of one another during the process of meiosis. So this incorporates genetic diversity as we're moving through. In terms of how we've been looking at inheritance of traits up to this point, we'd really focused around genes located on somatic chromosomes. Not somatic chromosomes, excuse me. Autosomes, that's what it is. The ones that are not your sex chromosomes. And so everybody has 2 pairs of those, and everybody gets one from mom and one from dad. But there are sex-linked genes. So these are genes that are technically located on the sex chromosomes, the X or the Y. The Y chromosome is really, really small and doesn't really have a lot of genes on it. The key one to note maybe is the SARY gene, SRY. That's the one that's responsible for making boys boys. But other than that, it's not of value in terms of a lot of genes. So when we talk about sex-linked genes, we're kind of automatically assuming that that means that that's a gene that's located on the X chromosome. And because females have 2 and males only have 1, it ends up affecting the sexes a little bit differently. The idea of dominant and recessive still applies, but it has to do with how many copies of it you have. And so in this case, eye color in flies is a sex-linked trait. So the wild type version is this red color, and then it's possible to then have a white color as a recessive or a mutation. Sex determination varies per organism. So we as humans have an XY system, but not all organisms do. On a previous AP test, we brought in the ZW system. I'm sure all of you are super excited about that. But that's for things like birds and roosters. Others have an XO system. So you either have 1 X or 2. They don't have a Y per se. And some of them are a diploid haploid system, which is kind of crazy. If you have 2 sets of chromosomes, you're a girl, but if you only have 1 set of chromosomes, then you are a male. So, sex-linked genes. As I mentioned, they're either on the X or Y, and you can see just from the picture, the Y chromosome is very, very small, and fathers determine the sex of the child. So they're either going to pass on an X chromosome to a daughter or a Y chromosome to a son. And since most of our sex-linked traits that we're talking about are going to be on the X chromosome, they get passed from father to daughter. And males only have 1 X, so they can be something called hemizygous. That means they either have it or they don't. They either have the good version on the X and they're fine, or they have the bad version on the X if we're talking about disease on the X, and they have it. They don't really have the opportunity to be a carrier. Females can be a carrier. What that means is they have 1 kind of healthy version of an allele and then 1 bad one, but the healthy one is enough to compensate. And since they have 2 X's, they have the ability to mask that disease that the males wouldn't be able to. So here's kind of an example of how a recessive trait might be passed via gametes. So in A, you have a affected male, and this is how we designate sex-linked traits. You're either XX as a girl or XY as a boy, and then the traits themselves in terms of what we would have done in Punnett squares previously with just regular genes, we put as a superscript. So there's still capitals and lower cases, dominants and recessives, and these are recessive traits or recessive diseases in this case, so they get a little letter. And the Y never gets a letter with it because we're not talking about a trait on the Y, it's on the X alone. So you create Punnett squares in a similar fashion. Every time you do a Punnett square, you would have half female, half male, because there's a 50-50 shot. And then you can assess whether or not those offspring would have the disease or not. So in this case, in A, you have 2 females and they are both capital and lowercase. So these would be an example of carriers. They wouldn't have the recessive disease because they do have a capital to compensate, but they do have the potential to then pass on that diseased X to future generations. And because mom was totally fine, all of the boys in the family are totally fine. And then here would be an example if the mom was a carrier and dad was fine. It shows you that you can actually have a male with the disease. So sex-linked traits are more prevalent in males, and you get them from your mom if you're a boy, because you get your X chromosome from your mom. And then it turns out likelihood increases if the male also has it. That's how you end up getting daughters that have the sex-linked disorder too, because they need to get an X from dad. So in order to have the disorder, traditionally, both dad has to have it and mom has to either have it or be a carrier. Typical sex-linked diseases include things like color blindness, muscular dystrophy, hemophilia. There's more. Another thing to kind of think about is something called Barr bodies or X inactivation. It's just something that happens in organisms too a lot of the time, because there is a, dosage problem. If you're thinking about, a male can survive and be fine with 1 X. Females have 2. So it's kind of like double the genetic material, and that can cause problems. If you think about Down syndrome, having a whole extra 21st chromosome causes a whole bunch of problems. So cells, what they naturally do is they do what's called X inactivation. They basically turn off one of the X's to prevent an extra gene dosage problem. And this happens in females during embryonic development. We just kind of turn one off and then we go with the other one. And it can be random inside the cells. So if you have, for example, a cat that has 2 different alleles, well then some cells are going to turn off the one for orange fur and some cells are going to turn off the ones for black fur, and then you end up with these different patches. It's kind of a female only thing, because males, they only have 1 X, so they're not going to turn it off, but females would. In terms of genetic recombination, we have talked previously about how traits assort independently of one another, and you can get all different types of offspring as a result. And we call the ones that look exactly like their parents parental types. So one parent was this and one parent was this, and these particular offspring look exactly like them. They have the exact same genotype. So they're parental types. And then we also have recombinants. Recombinants are the ones that have some traits from both parents. They don't have everything from one. They're a recombinant. And so because of that, there are instances where it might not follow the laws of independent assortment, and you don't get the necessarily like 9:3:1 ratio that you're going for in terms of parental types and recombinant types. And if that happens, then that's a suggestion that there are genes that are linked together. So if it's kind of breaking the traditional rules of probability, then most likely those genes are linked. What a linked gene is basically 2 genes that just happen to be super close together in terms of location on the chromosome. And when crossing over happens, which is how we get recombination in the first place, the likelihood is that they just travel together. They are so close that when a crossing over event occurs, it usually lumps the 2 together. And so you end up seeing way more parental types than you normally would in terms of recombination. Here's kind of just showing you what that might look like. If they do follow independent assortment, then they're not linked, but if they don't, they are. Oops, I'm going backwards, meaning to go forwards. So the idea here, just kind of rehashing, Crossing over takes place during prophase 1 in meiosis when tetrads are formed. So if two genes are far apart, then crossing over usually happens, and we consider it about 50% recombination. That's what these would be here. Whereas the ones that don't cross over, those would be parental types. The closer these two genes get together, then you decrease chances that crossing over will occur between them. They would have a lower recombination frequency, as it said. So there is a calculation for it. You kind of look at all of the offspring that occur, and you take the number that have successfully recombined or have some traits from both parents, divided by the total, and then you get a percentage. So in this particular instance, there was 17% that were recombinants. That would be called the recombination frequency. And that percentage is basically telling you how far apart those genes are. If you get 50%, then it's assumed that they are not linked at all. Anything under 50%, then now you're starting to get closer and closer together. And the lower percentage of recombination that you get means it's harder to occur. So they're even closer. So if we get 17% recombination, then it suggests those two genes are kind of 17% or one map unit, so to speak, apart. There's a couple other things that are kind of exceptions to Mendel's rule. There's something called genomic imprinting. And what that really is just kind of like a phenotypic effect occurs depending on if it came from a male or a female parent. In this case, I'm guessing you don't really need to know about this particular thing. In Unit 6, we learn about acetylation and methylation in terms of regulation of turning genes on and off. So that is something that can control genetics a little bit and then result in changes in phenotype. We have a ton of DNA, again, that's not in the nucleus. If we remember endosymbiotic theory, certain organelles have their own DNA, like mitochondria and chloroplasts. So mitochondrial DNA is passed from the mother to the offspring because you get all of your mitochondria from your mom. It's hiding in the egg. So if there's some kind of mutation in that mitochondrial DNA, then again, that can cause difference in genotypes and phenotypes as well. Other issues that can occur, one of the major ones that can result in chromosomal disorders like Down syndrome is something called non-disjunction. So that means when the homologous chromosomes line up in the center in tetrads, they do not separate as they should. So the homologous pairs do not separate as they would. Therefore, you end up in the first division with some cells that have less chromosomes than they should. So in this case, we started with 4 and we're ending up with just 1. Here, you're ending up with an extra chromosome that you didn't mean to have. So instead of 4 chromosomes going to 2 and 2, we now have 3 and 1. And that's what's kind of denoted here. You're going to end up with gametes that end up having an extra chromosome and some with a missing chromosome. If this egg, let's say, goes and gets fertilized by a sperm, that could be where Down syndrome comes from, if that extra chromosome is the 21st. One missing a chromosome might be something like Turner syndrome, where you're missing an X chromosome. Non-disjunction can also happen in the process of meiosis 2, and similar process, but instead of all 4 gametes being affected, only 2 would be affected, because kind of on the right here, everything is fine. So you can determine where the non-disjunction occurred based on how many gametes are affected. If half the gametes are affected, it must have occurred in meiosis 2. If all the gametes are affected, it must have occurred in meiosis 1. There's a ton of mutations that can occur on a chromosome. that are kind of more big picture. If a chunk of a chromosome gets deleted, it's also possible a chunk of a chromosome can have some repeats. A chunk of a chromosome can flip-flop itself. Sometimes there's something called translocation, where a chunk of one chromosome swaps places with a chunk of another chromosome. And all of these things can lead to, issues in the long run. And that kind of ties up the end of Unit 5. And then we're going to move into the chromosomal basis of inheritance.